start by, sorry about that. I wanted to start by seeing if there were any questions that anybody had that you wanted me to start with about anything that's going on with the class or any of last week's material, or any questions that anybody has. And if you've got questions, you can either type them in the chat box and they'll appear in the chat box, or you can do the raise your hand thing, click on the hands, and that'll tell me you have a question, and you can do audio if you have a microphone, either way. And then I want to see if anybody has any specific topics in these two chapters that you want me to go over. So have at it. What questions do you guys have? Is there anything in particular that anybody wants me to go over with this week's material, or anything in particular anybody wants me to start with? Okay, well, what I might do is kind of go through and talk about some of the topics that I think are more difficult to do on your own. Some of the structure stuff is just kind of memorizing parts and what they do, and that's going to be a little bit easier than some of the processes that are a little bit more difficult to understand on your own. So what I might do is go through some of those types of processes. As far as learning the organelles, I usually recommend to students that you make a chart to help you learn the organelles. In the first column, you can write the name of the organelle. In the second column, you can write something about its structure. Is it a membrane-bound organelle? Is it part of the, is it made of protein filaments? What's its structure? In the third column, I usually recommend that you try to relate the organelle to a macromolecule that we talked about in Chapter 2. And that'll help kind of fit it together with what you're already learning about. So, for example, mitochondria break down disaccharides and monosaccharides into energy molecules. So that fits kind of with the carbohydrate class of macromolecules. The rough endoplasmic reticulum makes proteins. The smooth endoplasmic reticulum makes lipids. So in that third column, just kind of link it to a macromolecule. And then in the fourth column, describe what its function is. So you've talked a little bit about what its structure is in the first column, and then in that last column you can talk a little bit about what its function is or what it does. It can be pretty simple. These can be single words or short phrases. This would just be a way to organize some of the information, and I think that might help you to learn about the organelles as you're going through. The other thing you can do is do note cards. If a table doesn't work for you, then maybe note cards. Put the name of the organelle on the front and some of that other information on the back, and then you can use those to study, and then you can use those to quiz yourself to see how well you're mastering some of that material. So that's one way to do the organelles. Now, one of the things that I want to talk a little bit about is the cell membrane. The cell membrane is sometimes also called the plasma membrane. It's a very flexible barrier. It separates the inside of the cell from the outside of the cell, but it's very strong, and it very much separates the interior environment from the exterior environment. It's sometimes called the fluid mosaic model of the cell membrane because it is very fluid. It's mostly made up of these phospholipids, but it's studded with proteins. And so these proteins can have lots and lots of different functions. But one function is they can act as gatekeepers to help certain ions get into and out of the cell. The lipid layer is a barrier to certain substances. And to think about that a little more, we can look at this particular figure. This is it's on page 43 of your book. It doesn't actually have a figure number because it's just embedded within the text. These are phospholipid molecules. Now, we talked a little bit, or your book shows you a little bit about phospholipids when it talks about lipids in Chapter 2. On page 32, figure 215, it shows you the figure of phospholipids, and we're going to look at that again in just a second. So we're kind of building again on information from the previous chapter. But the phospholipid is made up of these head groups, and those head groups are made of polar molecules, which means they're very hydrophilic. That means they like water and they like to be oriented in an aqueous solution. And then the tails are these fatty acid chains that we learned about last week. And we learned that fatty acids are very nonpolar. Or to use the other term, they're very hydrophobic. So you have this inner region that's very hydrophobic and this outer region that's very hydrophilic. And when you put them together, they form this arrangement where they form this bilayer of these phospholipids. And so this is the figure from this chapter that's showing the phospholipids, and it's showing you this hydrophilic head region and these hydrophobic tails. If we do this kind of molecular structure, you see these red molecules are oxygens, and that's what makes this portion very polar. And then the rest of it is just carbon and hydrogen. And so it's those very nonpolar hydrophobic tails. And so when we put a bunch of these molecules in a watery solution, most of our body fluids are aqueous solutions, these phospholipids very spontaneously line up in this way that these tails, these hydrophobic tails, are very happy because they're all surrounded by other lipids. And the water is being kind of protected from them or excluded by these polar head groups. And this figure does a good job of showing you this membrane stretches in 3 dimensions. This membrane stretches in all around the cell in kind of a 3-dimensional way. And so it tells you that this lipid bilayer spontaneously shapes itself kind of into a bubble. And there's an animation that shows you that kind of rounded shape that we tend to think about for cells. And so this structure of the cell membrane lends itself to a function that we're talking about called permeability. Now, your book talks about that structure and the function in 2 different parts of the chapter. So I'm actually skipping to another part of the chapter because I want you to connect the structure and the function. Your book talks about the structure of the cell membrane and then goes and talks about structures of lots of different other structures within the cell. And then it gets into some of the cell function, transport across membranes, making energy, some of those other kinds of things. So it really separates talking about the structure and talking about the function. But they're very much related. So I want to talk about them together. So I've kind of skipped a little bit later in the PowerPoint and a little bit later in the chapter. So based on that structure that we just talked about, cell membranes have a property that we call selective permeability. Cell membranes are selectively permeable. And that means that some substances can pass through the membrane and other substances can't pass through the membrane. It's based on that structure that we talked about with that interior layer of lipids. Because of that interior layer of lipids, anything that doesn't dissolve in lipid can't go through the membrane. It can't, so it's repelled by that inner lipid layer. And so that lipid bilayer is permeable. These are the things that can go through to gases such as oxygen and carbon dioxide. It's permeable to water, and it's permeable to small lipid-soluble molecules. But it's impermeable to anything that's polar, such as glucose, anything that's charged, like ions, and larger molecules. Now, when we do want some of these molecules, like glucose and ions, to get into the cell, we have proteins that can act like channels and transporters to get those substances across the cell. And we're going to do that as part of our process of cell transport. And so this is a figure from your textbook that shows you this selective permeability. They're showing you the lipid bilayer. They show you some things can freely cross that lipid bilayer. Gases, small lipid-soluble or nonpolar molecules, and water. Other things like larger molecules, glucose, ions, are blocked from crossing through. And so that's what we mean by selective permeability. So the structure of the cell membrane sets up selective permeability. And selective permeability sets up some of our transport processes. And so I want to talk about 4 types of transport processes, 4 ways that substances get into and out of the cell. The first type of transport is diffusion. And diffusion is just movement of a substance from an area of high concentration to an area of low concentration. Molecules like to spread out, and so diffusion takes advantage of that and helps molecules to spread out. So a lot of times when I'm teaching this topic in class, this kind of goes back to some of our vocabulary about solutions that we talked about. And so I often tell students to kind of think about a solution that most people are pretty familiar with, which is Kool-Aid. It's the time of year when lots of people are making Kool-Aid. So normally in class, I'll ask people to think about how we make Kool-Aid. Kool-Aid is actually an aqueous solution because we take solute molecules and suspend them in water. We take the packet that is the color and the flavor molecules, and usually you take a cup of sugar. Those are your solutes, and you're dissolving those in water. So Kool-Aid is an aqueous solution. And so then I ask people to think about what happens if you make Kool-Aid and you add too much water. And most people come up with terms like it's very watery or it's very weak. And so we talk about how that is a solution that has a very low concentration of solutes. Because there's so much excess solvent, the concentration of solutes in that solution is very low. So the watery Kool-Aid or the weak Kool-Aid or the dilute Kool-Aid is an example of what we're talking about when we're talking about a low concentration. And then I usually ask people to think about what happens if you add too little water when you're making your Kool-Aid. Usually people say it's gross. But a lot of people will use the term strong. Your Kool-Aid is too strong. And what we're talking about there is a solution that has a high concentration of solutes. And so what we're thinking about when we think about this idea of diffusion is we're thinking about molecules wanting to spread out, to move from an area of high concentration to an area of low concentration. And so that's what we mean when we're talking about these terms like concentration and concentration gradient and diffusion. A concentration gradient is just the difference in concentration between two areas. So when you're making the Kool-Aid and you dump the packet in the bottom of the pitcher and you dump the sugar in the bottom of the pitcher and you first put the water in before it fully dissolves, those molecules are at a high concentration in the bottom of the pitcher and in lesser of a concentration as you go through the water. To make the solution, you stir it so that all those molecules spread out evenly throughout the solution. And so that kind of helps make this slide make a little bit more sense. So there are different things that we do to help substances dissolve. One thing that we can do is that we can stir things, and that'll reduce diffusion distance. When you stir it, you're reducing the distance that it has to diffuse. Another factor that affects rate of diffusion is temperature. And if you drink coffee or you drink tea, you've recognized that sugar dissolves in hot coffee and hot tea much more readily than cooler coffee and tea. And so temperature can affect the rate of diffusion. So these are some of the factors that can affect the rate by which something diffuses through a solvent to become a solution. The second type of transport that we're talking about is osmosis. And this definition of osmosis is a little technical until we kind of get into our discussion of it. So it's the diffusion of water across a semi-permeable membrane or a membrane that has selective permeability, like the cell membrane, in response to solute differences or a concentration gradient. And so once we go through our discussion, that definition might make a little bit more sense. And the first thing I always try to do is talk about some terms. There's a lot of terminology that's in this section of the book. And I always want to try to make sure that everybody understands terms pretty well. So one term that you'll hear is hypotonic. Another term that you'll hear is isotonic. And the third term that you'll hear is hypertonic. And I want to kind of define these a little bit. Tonicity is a measure of the concentration of the solution that's outside the cell. So it tells you how much solutes you have outside the cell. So whenever we're talking about tonicity, we're going to be talking about outside the cell relative to inside the cell. The prefix hypo means less. And so the hypo part tells you less, and the tonic part tells you you're referring to the solutes outside the cell. Hyper is a term that means more or greater than, so you're talking about more. And again, the tonicity tells you solutes outside the cell. We're referring to outside the cell versus inside the cell. And then iso means equal or same. So here you have the same amount of solutes. outside the cell. Usually the solutes that we're talking about in osmosis are the solutes that cannot pass through the membrane. They can't go through that selective permeable membrane. So we're talking about things like sodium ions or chloride ions or potassium ions. Or maybe we're talking about glucose or sucrose. So those are the type of solutes that we're talking about. Now they can't pass through the cell. So even though we have a concentration gradient in this hypotonic, we have less solutes outside the cell than we do inside the cell. In hypertonic, we have more solutes outside the cell than inside the cell. Even though we have that concentration gradient going on, that solute can't move across the membrane to balance that out. Now, our things that could move were gases and lipid-soluble molecules and water. And so what we need to do is we need to know about the water, because the water is what can move in order to balance this situation out. So again, the hypotonic solution is like our weak Kool-Aid. It has less concentration of the Kool-Aid. It has less concentration of the solutes because we eat too much water. So here there's more water. And again, we're talking about outside the cell relative to inside the cell. So if you have less solutes, you have more water. In the hypertonic situation, it's going to be the opposite. This is like your thick Kool-Aid, your gross Kool-Aid. You didn't add enough water for the solutes that you have. So you have more solutes and you have less water. Again, outside the cell. We're always talking outside the cell relative to inside the cell. Now, luckily, for isotonic, the same amount of solutes outside the cell, it means you have the same amount of water outside the cell. So the isotonic situation is a balanced situation. Hypo and hypertonic are out of balance, either one way or the other. Hypo, less solutes. Hyper, more solutes. So now that we know what these terms mean, we can look a little bit at what happens to a cell in each of these situations. So we can look at the hypotonic situation first. So we're going to look at what happens to a cell when we put it in a hypotonic solution. And again, this hypo means less, so this is less solutes outside the cell. And so that means that we end up having more water outside the cell. So we can draw a cell. For the example for the solutes I'm going to use sodium. And so here we have less sodium ions outside the cell than we do inside the cell. But sodium ions are charged. They're not lipid soluble. They can't go through that lipid membrane. So even though they would like to spread out, and even though they would like to be evenly dispersed across this membrane, the membrane is selectively permeable, and these are one of the things that is prevented from going across the membrane. So the only thing that can move to balance out the situation is water. And so we have to figure out where water is, and there's more water outside the cell than there is inside. And we know that because of what we know about the definition of hypotonic solution and what it means. Osmosis is the movement of water down its concentration gradient. So water is in higher concentration outside the cell here than inside. So water will move into the cell to even out. This cell is going to swell up, and if the osmotic gradient is great enough, if enough water moves into the cell, this cell might burst. And if that happens to a red blood cell, that's called hemolysis, lysis of the red blood cell. Lysis means splitting apart or breaking open. So we can look at the same thing for a hypertonic solution. A hypertonic solution, hyper means more. Tonic means solutes outside the cell. But again, we're going to need to figure out where the water is. If there's more solutes, there's less water outside the cell. So we can draw ourselves a cell. Again, using sodium, here there's more solutes outside the cell. So we're going to have much more of the sodium outside the cell than we had inside the cell. We're going to have less water. And that means there's much more water inside the cell. And this is all just based on what we know about how solutions form, based on the definition of the type of solution we're looking at. So we know how solutions form. Solutes in a solvent. And now we've learned that if you look at this prefix, that tells you about the concentration of solutes in the solution. And so we can figure out where the water is. So here the water is in high concentration inside the cell. So when the water moves to balance out the concentration gradient, because the sodium can't move because this is a selectively permeable membrane, here this cell is going to shrivel up because water is moving out of the cell. And so the cell will kind of shrivel up because there's less water inside of it. And if that happens to a red blood cell, that term is called crenation. And so that's kind of demonstrated to you in this figure in your textbook. They show you kind of this bag that's supposed to represent a cell. And it has 2% sucrose in it. And this bag is a membrane that is permeable to water, but is not permeable to sucrose. When you put it in a situation that is the same on the inside and the outside, this 2% sucrose is an isotonic solution, you won't get any net movement of water. But if you put it in a 10% sucrose, 10% has 5 times more solutes in that extracellular solution. So the water is going to move out of that bag, and that bag is going to shrivel up. If you put that 2% sucrose in water, which is 0% sucrose, now it's a hypotonic solution. And now the cell is going to swell up because water is coming into it. And your book shows you the same figure using red blood cells in an isotonic, a hypertonic, and a hypotonic solution. So this is one of the reasons when you give someone intravenous fluids, you don't give them just plain water. You actually usually give 0.9% saline because that's isotonic with the inside of red blood cells. If you use just plain water, then you'll get the hemolysis. There's not enough solutes. Water will move into the cells, and the cells will swell up and sometimes even burst. If you use too much saline, then it's a hypertonic solution, and the red blood cells will crenate. That means they don't carry oxygen as well, and they don't move through the blood vessels as easily. And so this figure kind of gives you a summary of what we've talked about. What I gave you is kind of the mechanism for how it happens. And there are several animations that are in the PowerPoint and also listed in a folder in the module that you can take a look at. The third type of transport that we're talking about is facilitated diffusion. Facilitated diffusion is still diffusion. So it's still going to be diffusion of molecules down their concentration gradient. But this time, this is the movement of ions that can't get across the membrane by themselves. The ions, the glucose, the bigger molecules. And so there's a protein that helps or facilitates those molecules getting across the membrane. They're still moving down their concentration gradient, but this time a protein is helping them. So we first talked about diffusion. Diffusion is just substances diffusing across the membrane down their concentration gradient, not using a protein or anything else. So that was only for gases and lipid-soluble molecules. Then we talked about osmosis. Osmosis is water balancing out concentration gradients. This way, sometimes you actually do need glucose to get into the cells. Sometimes you actually do need ions to get into the cells, not just to balance out a concentration gradient. Water can do that for osmosis, but actually to get that molecule inside the cell. And so this is a way to get that molecule inside the cell. Your book sometimes refers to this in the animations as passive transport, because although it uses a protein, it's not using any energy by the cell. The substance is simply moving concentration gradient. So again, they show you the lipid bilayer. They show you a protein here. So now this can help glucose get past that lipid bilayer and get into the cell. But glucose is still going down its concentration gradient. So this is just a type of passive transport. We're not using any energy by the cell. Now the fourth type of transport does use energy. It consumes ATP. ATP is one of the ways that our cells use energy. And because of this, it can transport ions independent of their concentration gradient. We sometimes call those ion pumps because they can pump things against their concentration gradient. So now we're no longer a slave to that concentration gradient. Instead of just letting molecules spread out because they want to, now we can confine them to an area of the cell because we're using energy. We're putting energy into that. And one example of that is the sodium-potassium pump. This shows the sodium-potassium pump. The sodium-potassium pump moves 3 sodium out of the cell for every 2 potassium ions that it brings into the cell. And that's what they're showing you here. They're showing you 3 sodium ions with the use of energy being moved out of the cell and 2 potassium ions coming back into the cell. So this actually sets up 2 concentration gradients. It sets up a lot of sodium outside the cell, and it sets up a lot of potassium inside the cell. And this is going to be required for how muscles contract and how nerves function. This is going to be important for the way a lot of cells have their function, the way a lot of cell functions take place. And there's several options that show active transport and the sodium-potassium pump that are on Blackboard and in the PowerPoint. So I want to kind of summarize a little bit some of the types of transport that we've talked about. We've talked about diffusion, osmosis, facilitated diffusion, and active transport. And I recommend that you might want to summarize some of your material like this, making a table. In the first column, we can talk about what moves. In the second column, we can talk a little bit about what direction. I'll tell you what I mean by that in just a second. We're not talking about what direction across the membrane, like inside or outside. We're talking about concentration gradient direction. We can talk about, does it use a protein? And we can talk about, does it use energy? And so we can kind of summarize our 4 types of transport looking at these 4 aspects. So the first type of transport we talked about was diffusion. Diffusion moves the solute molecules, and it moves them down their concentration gradient. So these are molecules that are trying to spread out, and these are molecules that can move across the membrane. Does not require protein, does not require any energy. And then the second type of transport that we talked about was osmosis, and osmosis moves water down its concentration gradient. That's actually going to be the opposite of the solute gradient. The solute gradient might be high outside the cell and low inside the cell. Water will be high inside the cell and low outside. So a lot of times you'll hear the moniker, a lot of people repeat this, water follows salt. And what they really mean is water follows the solute. And that's a way to explain osmosis. Osmosis does not require the use of a protein because water can freely cross the membrane, and it does not require energy. It's a type of passive transport. Now, sometimes we actually do need to get certain solutes across the cell. We actually do need glucose in the cell, not just to balance out a concentration gradient with water, but actually because we need glucose into the cell. And so we can let glucose move into the cell down its concentration gradient if we provide it with a protein. Now we're starting to use a protein to get things across that membrane. Kind of acts like a drawbridge over a moat, or just kind of opens up a pore or a channel to let things go through to kind of protect them from that inner lipid layer. But because we're still going down the concentration gradient, we're not using any energy. And then active transport also moves solutes, but it doesn't care about the concentration gradient now. Now we can move things against the concentration gradient because we're using a protein and we're using energy. So this is a way to kind of compare some of the different types of transport that we talk about in this particular chapter. Now, sometimes you need to get lots and lots of things across the membrane, and that's where membrane transport or transport in vesicles comes in, and that's also kind of described in your chapter a little bit. Does anybody have any questions on any of that? Some of those types of transport? Does that help at all? Does that kind of clarify anything based on your reading? Okay, good. I have heard that the Kool-Aid thing helps to put it in perspective because we're talking about solutions, and you can kind of understand when we talked about solutions in Chapter 2 as vocabulary, okay, that's something dissolved in something else, okay. And if it's dissolved in water, that's an aqueous solution. But then as we actually really start to talk about solutions in this chapter, having a more concrete idea about that in your head really helps to kind of give you an image to know what we're working with. And most of the things that we eat and drink are aqueous solutions. We just never think about them in that way. And so using something that's common like that, I think, is pretty helpful. And I use tables a lot to organize information. I think they're really helpful. And there's a lot of times when you're studying and you're going through something in the book, you should always read with a pen or a highlighter or something in your hand. But sometimes it's good to just kind of stop and go, okay, how can I organize this information? And sometimes they don't work, but sometimes tables are a good way to kind of organize some information and to summarize it. And then you can use that to kind of help you study rather than trying to carry around a whole chapter in your book. That kind of helps you distill down some of the information too. So I tend to use tables a lot. That's why I tend to suggest sometimes tables being helpful. A lot of that sometimes depends on your learning style too, but sometimes they're helpful. So I want to talk a little bit about some of the things with energy in this chapter. This chapter is really setting up Chapter 5, which we are not actually going to have a lot of time to go through this summer, so I actually kind of skipped Chapter 5 in your syllabus. So what they're doing here is setting up some of the information for Chapter 5, because Chapter 5 talks about metabolism. Chapter 5 actually talks about how do we take energy out of food and make it into energy that our cells can use. And so in order to understand that, they give you some kind of basic concepts about energy. And so one of the things that I always say when I teach this class is when we're hungry, we don't go sit in the sun. We eat a plant or we eat an animal that ate a plant, because we do not have the capability of taking sunlight energy and making it into cellular energy. Plants can take sunlight energy and can make it into like glucose. And we can take glucose and make it into cellular energy like ATP. But we can't do that directly from sunlight. So when you're hungry, you can go sit in the sun all you want to. You will still be hungry. Your cells will still need glucose. So when you're hungry, you eat a plant or you eat an animal that ate a plant, and that gives you the raw materials your cells use to make the energy in the way that they can use it. And in the way that they can use it is usually a molecule called ATP. So what we're doing is we're trying to provide energy for the cell to do work. And what we mean by work is things like moving ions across the membrane. We just talked about active transport, and we said that requires energy because you're pumping ions against a concentration gradient. You're kind of rolling a ball up a hill there, and you have to have energy to roll a ball up a hill. You have to have energy to pump those ions against their concentration gradients. And so that requires energy. And so when your book talks about doing cellular work, those are the kinds of things it's talking about. It's talking about moving ions. It's talking about contracting fibers, like in muscle cells, to contract a muscle. So those are the kinds of things that it's talking about cell work. Some cells, like bacterial cells, have flagella, which are little tails. So it requires energy for those tails to move, for those cells to move. So that's another example of cell work. And so your book goes into some of the details about some of the energy laws, some of the laws of thermodynamics. And this one, the first law, is kind of relevant when we study metabolism. Energy cannot be created or destroyed. It can just be transferred from one thing to another or converted from one form to another. And so here's where I put it in the slides. When we're hungry, we don't go sit in the sun. We eat a plant, or we eat an animal that ate a plant to get the food energy. So the plants can convert energy from the form of sunlight energy to the form of food energy. And we can convert it from the form of food energy into cellular energy. We're not creating energy from scratch. We're converting it from one form to another. And so that's kind of what they're saying by that first law of thermodynamics, is we don't just spontaneously make cellular energy. We have to get it from the food that we eat, from glucose that's found in plants and animals. And they go through and they talk a little bit. They talk a little bit about the fact that we can store energy in molecules. We can actually store energy in chemical bonds. So the chemical bonds that we talked about in the last chapter, we can actually put energy into those chemical bonds. And so we formed those chemical bonds by performing chemical reactions. We formed covalent bonds, we formed ionic bonds. Most of the time we're talking about covalent bonds when we're talking about storing energy in bonds. We're talking about electrons sometimes when we're talking about energy. So a lot of times we're talking about the covalent bonds. And so when we put molecules together, or we put atoms together, what we start with is often called a reactant that goes into the reaction, and what we end up with is often called a product. And so when we're writing this as a chemical reaction, we write whatever the reactant is in front of an arrow to show that a reaction has taken place, and then we write the product on the other side of the arrow. And sometimes reactions are reversible. So it just kind of depends on what the conditions are, which direction it goes. And so they show you an example. This is on page 64 in your book, and they show you an example. They show you the reactants and the arrow and the product. And we've not changed anything. We still have 2 hydrogen. We still have our oxygen, that O and that 2. And so we still have the same number of hydrogen atoms and oxygen atoms as we did. Rather than being hydrogen molecules and an oxygen molecule, they now form these water molecules. And so that's an example of bonds forming, those covalent bonds forming. So this is, again, still trying to build on what we were talking about in the last chapter. And so chemical bonds can hold energy. And we can cells can store energy in these chemical bonds. And specifically, the way that it tends to do that is in a molecule called ATP. ATP stands for adenosine triphosphate, but we normally abbreviate it ATP. The T means that there's 3 phosphates. And so to take a look at the ATP molecule, here's showing you all the atoms in it. But what they're showing you is that it has these 3 phosphate groups. And so ATP has 3 phosphates, and the bonds that hold that phosphate on have a lot of energy in them. And if we break that bond, that energy can be released. So that ATP has 3 phosphates. We can break that bond, and that will release energy that can go to do cell work, or that can be used in other reactions that require energy. And so what we'll be left with is an A with 2 phosphates, adenosine diphosphate, and this phosphate just kind of running around. If we can combine those with more energy, then we can put them together to make ATP. And so this is a reversible reaction. ATP can be broken down into ADP plus a phosphate plus energy. Or we can put energy and a phosphate and ADP back together to form ATP. And so this is what your cell does. What I've shown you in the orange is how we make ATP. That happens in the mitochondria. In learning about the mitochondria this week, you might have come across it's the powerhouse of the cell. It makes energy. It makes ATP. That's what it does. It takes energy from the food that we eat, combines ADP and a phosphate with that energy to make ATP. And then that ATP can go all over the cell and be used for cell work. And so what I showed you with the blue arrow happens, for instance, when we move ions across the cell, or when a muscle contracts, or when the flagella helps the bacteria to swim. And so it's this cycle. It's this never-ending cycle. It doesn't all happen in the same place in the cell. So it's not like you're spinning your wheel. The ATP is made in the mitochondria and then goes other places in the cell. And then those components can come back to the mitochondria and pick up more energy and then go other places in the cell and then come back to the mitochondria and pick up more energy. And then your book also talks a little bit more about enzymes, because enzymes can catalyze these reactions. And whenever we have a chemical reaction, if it includes an enzyme, the name of the enzyme is written above the arrow, because the enzyme doesn't actually participate in the reaction. It's not changed. It can go on to do other reactions. And so this next part of your chapter talks about enzymes and how enzymes work, because in chapter 5, it kind of ties all this together to talk about how plants make glucose and then how we metabolize glucose and turn it into ATP. So it's trying to give you some of these basic ideas about ATP can store energy and enzymes can catalyze reactions, because the whole next chapter is about the reactions that help to make ATP. And so you're welcome to take a look at that chapter if you want. We're actually not going to have time to cover it in a lot of detail, so it's not anything that's required. If you're a big picture kind of person, that might help you understand some of these parts of the chapter that seem really isolated. Okay, it talks a little bit about energy. Okay, now it's talking about enzymes. And then, you never get to the so what, because the so what is in the next chapter. So I'm hoping that kind of explains a little bit about that part in the middle about energy, because what they're kind of getting at in this chapter is cell structure and cell function. And one of the functions of cells is to make ATP. One of the functions of cells is to transport things across the membrane. So they give you a lot about structure, and then they give you a little bit about those functions. No, nothing from Chapter 5 will be on the test. The test is only going to cover Chapters 1 and 2 and Chapters 3 and 4. Nothing that you will be graded on will come from Chapter 5. That's a good question, Nicole. Okay. Other questions that you guys have? I haven't actually designed the test. I can give you a little bit more information a little bit later in the week, and I'll probably post some kind of an announcement to kind of give people an idea. It'll be some multiple choice. It'll be a little bit of fill-in kinds of questions. It's just going to ask some very general kinds of questions about some of the things that we're talking about as we go through the PowerPoints. So if you take a look at your PowerPoints and use those kind of as a study guide, then you'll probably be in good shape. It's not going to be a lot of recall because there are certain things that if you need to know them in life, you can go look them up in a book. And so I'm more interested in are you kind of piecing together some of the things that we're learning, especially because some of these chapters really kind of relate one on top of the other. We're still referring to some of the stuff we did in Chapter 2 as we talk about Chapter 3. We're referring to structures in Chapter 3 as we talk about Chapter 4. So I may ask you kind of some overall things like describe a function of the cell. And I'm not looking for a lot of detail. I'll tell you if I'm looking for specific details or those kinds of things. But I'm more looking for can you relate, do you know in general things that cells do? For instance, they make energy. For instance, they transport things across the membrane. I could ask you what are some ways that the cell gets things across the membrane without using any energy? And so you could talk about diffusion or you could talk about osmosis. I'm not looking, I'll usually tell you if I'm looking for a big lengthy answer. Usually I'm just kind of going to kind of look for short types of answers. Then Chapter 1, Chapter 2, Chapter 3, Chapter 4. As far as length, I'm really not sure about what the length of it's going to be yet. Again, I was going to kind of work on that and then put together kind of a guide that I was going to post for announcements to kind of give people an idea when we got a little bit closer to time. The test will be available the times that are listed on the syllabus. You will only be able to access it once. So you're going to want to make sure that you're ready when you get ready to access it. The test is designed to be pretty short. It's only going to be 20 points. So it'll probably have between 15 and 18 questions. Most of them will only be worth a point. Maybe there will be a couple that'll have. Yeah, I think you are on the right track, Robin. Robin's talking about kind of learning the basics of the information, and then that'll provide the basis for other information. So the idea, I think, is that we're going to try to build things on as we're going chapter by chapter. There's a lot in each of the chapters, and I'm hoping that I'm kind of helping you focus on the things that are most important. And I'm trying to do the things that are going to reappear in later chapters. So we're going to get into talking about DNA, replication, transcription, translation. That's going to build on structure of the nucleus. So I'm trying to focus on the things that are going to be recurring themes in other chapters and not just esoteric details that if you ever need to know them in the future, you can look them up in a book. Because my dad still does that. My dad loves to read science news, and then he doesn't understand something, and he'll come dig out one of my textbooks to go and look something up. So, the goal is not that you understand all of the little details, but that you understand enough about processes that if you go look up the details, that you understand what you're reading, and that you know kind of keywords to use if you want to look up something. So that's my goal, hopefully. Other questions that you guys have? Okay, good job, you guys. I'm going to work on getting the discussion board finished grading and the assignments graded. Probably the discussion board will be graded in the next day or two. The assignments might not be graded until the end of the week. So I'm going to take a look at those and kind of let people know what I think about those. So you'll start to see some stuff appear in the grade center over the next few days is my hope. And again, if there's ever any questions or anything, I think that questions on the discussion board is working pretty well. So, you can get to the Grade Center through the Tools tab, but yeah, I can put a My Grades tab up so that all you have to do is click on the My Grades and you can get there. The assignment is, each assignment is under the Module tab. So if you go to Module 1, you'll see the Module 1 discussion board, and it links you directly to the discussion board, and you'll see the Module 1 assignment. If you click on that, that's where you download the sheet that explains the assignment, but that's also where you will upload your assignment. And the Module 2 tab is the same way. It has, under that Module 2 folder, there's a link for the assignment. That's where you can go to download the assignment sheet that explains the assignment, and also where you will upload when you've got your assignment written. So. There's actually two things that you'll work on each week. You'll actually work on the discussion board and you'll work on the assignment. The discussion board has a prompt, and so you'll prepare an answer to that prompt and put it on the discussion board, and everyone can see and everyone can respond to that. And the syllabus has the requirements for that grading. Your response needs to be at least 250 words. You need to respond to at least 3 of your colleagues and participate in the discussion. Talk about how what you're learning relates to what they said. So that's the discussion board. And then the assignment is one that you will work on and that you'll type up an answer to as like a Word document and upload and turn in. And I'm the only one that sees that, and then I'll do grading based on that. And the grading criteria for both of those are listed in the syllabus. So it gives you kind of an idea of what I'm looking for the discussion board when you look in the syllabus. So I know that that's, I tried to organize it so when you looked at the module, you could see, okay, there's the Limba classroom, and there's the PowerPoint, and there's the discussion board, and there's the assignment. But I know that that's kind of been a little bit confusing for some people. I didn't put a word limit on the assignments. Some of them are going to be longer than others. The 250 words is for your initial response to the discussion board. So I just didn't want people to give me kind of a 5-word sentence as their answer to the discussion board. So that's why I did the word minimum on the discussion board. There's not any kind of a word guideline on the on the assignments. Some of them are going to be longer than others. So I didn't really give a guideline for the assignments. Okay, well, I'm going to go ahead and turn off the archiver so that I can make sure the archive is available to everybody. And I'm going to put up the My Grades button because I didn't even think about that not being up there. And I'm going to work on reading some more discussion board posts and some assignments. So you guys have a good afternoon.